In the last class we have seen the requirements for ATP synthesis and we have discussed about chemi-osmotic hypothesis. Now let us see how ATP synthesis occurs in mitochondria. So this is the mitochondria and we know that ATP synthesis it occurs inside the mitochondria. And mitochondria is also a double layered membrane. This is the outer membrane and this is the inner membrane. And this space, these are the cristae, and this space is called the intermembranous space. Intermembranous space. Once again I will tell you, this is the outer membrane, this is the inner membrane and this is the intermembranous space, and inside this space protons are accumulated. So we have seen in chloroplast where the protons are accumulated inside the granule thylakoid, inside the lumen of the granule thylakoid. But in mitochondria the protons are accumulated inside the intermembranous space, and the protons they come out through the transmembrane particle, that is F1 F0 particle. This is the head and this is the channel. When the protons they come out into the matrix, this head turns and ATP is synthesized in the matrix. So there we have seen in chloroplast what happens. The ATP is synthesized in the stroma, and from the granule thylakoid the hydrogen ions they come outside into the stroma. Here the protons are accumulated in the intermembrane space, and this is the channel F0 F1 channel, and through this channel the protons they move out of the membrane and hence ATP synthesis occurs. So in mitochondria oxidative phosphorylation is occurring, that is ATP synthesis is occurring with the help of oxygen, and in chloroplast photophosphorylation is occurring, that is oxygen synthesis, my ATP synthesis it is occurring with the help of sunlight. So I think you have understood. So please go through the previous video. Hi friends, we have seen that for ATP synthesis the concentration of protons has to increase in the granule thylakoid of the chloroplast. So this is the chloroplast. This is the structure of a chloroplast. And this whole structure is the granule thylakoid. This whole is the structure. This structure is called the granule thylakoid, and we have seen that this is the lumen or the empty space of the granule thylakoid. So for ATP synthesis to occur, the concentration of protons has to increase inside the granule thylakoid. Now let us see how the protons increase in the granule thylakoid. Now here this is the photosystem 2 and photosystem 1. So let us see, and this is the stroma, and connected to the lumen are the channels F0 F1 channels. Only if the protons come out of the channels, then only ATP is synthesized in the stroma. So let us see how this proton ion concentration increases in the lumen of the thylakoid membrane. Hydrogen ions the protons has to increase inside the lumen of the granule thylakoid, then only ATP synthesis will take place. So how this happens? First of all, this is photosystem 2, this is photosystem 1 in the granule thylakoid. In the photosystem 2, what happens? Photolysis occurs. What is photolysis? Breaking of water. So breaking of water or photolysis it occurs in the lumen of the granule thylakoid. So water is broken and oxygen, protons and electrons are made. So the concentration of protons increases in the lumen of the granule thylakoid. So photosystem 2 it carries on the photolysis of water, breaking of water, oxygen, protons and electrons are made, and hence the concentration of protons increases inside the lumen of the granule thylakoid. So the first method by which protons increase inside the lumen of the granule thylakoid is photolysis. Now the second method of increasing the concentration of protons in the lumen of the granule thylakoid is decreasing the concentration of protons in the stroma. How will the concentration of protons decrease in the stroma? We know that photosystem 1, it makes NADPH out of NADP plus. In photosystem 1, the electrons are given to NADP plus, protons are also given and NADPH is made. So this process takes place in the stroma. So what happens? The concentration of protons, it decreases in the stroma. So second method is to decrease the concentration of protons in the stroma. In the photosystem 2, it gives the electrons to NADP plus and it makes NADPH. So protons, the number of protons reduce in the stroma, concentration of protons reduce in the stroma, decreases in the... Now the third method of increasing the concentration of protons inside the lumen of the granule thylakoid is that we know that in the light reaction, electron is moving from photosystem 2 to photosystem 1 and in between there are many electron carriers. So photosystem 2, it gives its electron to pheophytin, which is the primary electron acceptor. Photosystem 2, it gives the electron to pheophytin and pheophytin, it gives the electron to plastoquinone. Plastoquinone is a hydrogen carrier. It is an electron plus, it is not an electron carrier, it is a hydrogen carrier. So it will bring the electron plus hydrogen into the lumen of the granule thylakoid. Plastoquinone will bring the proton into the lumen of the granule thylakoid. Once again I will tell, we know that during light reaction, electron is transferred from photosystem 2 to photosystem 1. From photosystem 2, the electron is given to pheophytin and pheophytin gives the electron to plastoquinone. Plastoquinone brings, it is a hydrogen carrier, it brings the hydrogen into the, it releases the hydrogen into the lumen of the granule thylakoid, hydrogen ions. So the number of protons or hydrogen ions, it increases inside the lumen of the granule thylakoid. So we have seen the three methods by which the protons can increase in the lumen of the granule thylakoid and the protons, they decrease in the stroma. So only then if the number of protons it increases in the granule thylakoid, then only what will be made? ATP synthesis will take place because protons will come out through the F0 F1 channel. The head of the F1 channel will turn, which is known as conformational change, and ATP is synthesized by using this energy. ADP combines with phosphate and ATP is synthesized. So we have seen the three methods by which we can increase the proton concentration inside the lumen of the granule thylakoid.